So, good morning, dear students. Today, we'll be commencing about the topic of photosynthesis. Photosynthesis. So, in presence of light, food is being synthesized. A very important aspect of the ecosystem itself is the producers or plants. If the plants do not exist, there can never be an herbivore. If there is no herbivore, there cannot be a carnivore. Life itself couldn't exist if producers aren't there. See, producers, the plants, they are also liberating oxygen during photosynthesis. So one of the important aspect, none of the things that we have invented have that capability to do what a photosynthesis does. Okay, what is the plants doing it? They are capturing the solar electromagnetic radiation, that is SEMR, see of nearly whatever sunlight that it falls. So the wavelength of light that could be utilized by the plants is very less. So only 1% of the photosynthetically active radiations is taken up by the green plants. And green plants, in the sense, they are green in color because of the presence of chloroplast. So chloroplast, if you have to say that, I can say it as if it is the Annapurneshwari for the entire ecosystem. That is, it is the thing which synthesizes food for the entire ecosystem. See, the chloroplast, the chlorophyll pigments in the chloroplast, they are able to absorb solar electromagnetic radiation. So they capture, conserve, and convert the solar electromagnetic radiation into food molecules. That is, they are converting this light energy into uh, chemical energy. So that is what the plants are doing it. So for the formation, see, two raw materials, if you take in a lab carbon dioxide and water, so and if you're going to fuse them in the lab, you're going to get carbonated water or soda. You're not going to get glucose there. Okay, for that molecule of glucose to be formed, so they should undergo photosynthesis in plants. Okay, so it's a complicated process, the photosynthesis process, which we'll be discussing in detail in the present chapter. So the green plants, they are able to capture, conserve, and convert the solar electromagnetic radiation into chemical energy due to the presence of chlorophyll pigments in the plants. So they do that job. So they are going to sustain the energy that has to flow along the various trophic levels. See, in the next year, next academic year, you'll be studying about ecosystem, the flow of energy from sunlight to the plants, from plants to the primary consumers, secondary consumers, tertiary consumers. The flow of energy will also be studying about. So it's an important aspect that a plant does. And for the entire life to sustain on Earth, the presence of plants is very, very important. So we cannot ignore it, and life cannot survive without the plants too. So we have to remember about the importance of this plants in the ecosystem and the process, that is photosynthesis, which we have not yet been able to replicate in the labs. Okay, what the plants are doing in the chlorophyll pigments is a unique phenomena in the ecosystem. And photosynthesis is sustaining life on Earth. Majority of life on Earth is sustained on Earth because of photosynthesis. There are chemosynthetic organisms also, but major role played by life for life to sustain us through photosynthesis. That is a very, very important process that is taking place in nature. So let us understand about this process, and step by step we'll be understanding it before we proceed into the uh, various types of reactions in the photosynthesis process. We'll also study the historical discovery of how they discovered that plants are liberating oxygen. Okay, light is necessary for plants to liberate oxygen. So many experiments were conducted, and also how food is synthesized only if carbon dioxide is taken up. So these many experiments will be discussing about in detail of it. So in this chapter, We'll study the historical aspect of photosynthesis. We'll also be studying about the mechanism of photosynthesis, like you'll be studying about light reaction. Under light reaction, you're going to study two types: cyclic photophosphorylation, non-cyclic photophosphorylation. So non-cyclic photophosphorylation is the one where we should concentrate more on. Then there is dark reaction, that is, it is light independent reaction. Dark reaction is nothing but light independent reaction. So dark reaction, the Kelvin cycle, we would be studying about where there is carboxylation, that is, carbon dioxide is fixed for the first time in the dark reaction or Kelvin cycle. C3 cycle, we also call it as. After that, we would also be studying about C4 cycle, the Hatch and Slack pathway or C4 pathway. So after the C4 pathway, we'll be studying the factors which affects the rate of photosynthesis. So these are the topics that we'll be covering under the photosynthesis chapter. So let us proceed with the historical aspect of the photosynthesis. So you can see that photosynthesis is a physico-chemical process. It's a physical as well as a chemical process by which green plants use light energy, that is solar energy, to synthesize organic compounds. See, the major raw materials for photosynthesis is carbon dioxide, water, and carbon dioxide is obtained through the stoma, and water is obtained through the xylem. So by the transpiration pull theory, you know about that. So carbon dioxide and water, so they are going to fuse in presence of light energy, in presence of chloroplast, and they are going to form glucose. In the process of photosynthesis, oxygen is liberated. So we should understand about that. So 6 carbon dioxide plus 6 water, it is in presence of chlorophyll and a photon of light, h nu. So we are going to get glucose plus 6 oxygen plus 6 water molecules. So 6 oxygen is liberated. uh, Diatomic molecule of oxygen is liberated during this photosynthesis process. So photosynthesis is a physico-chemical process by which green plants, they are going to use light energy or solar energy to synthesize organic compounds. So photosynthesis is the basis of life on earth. So it is the very existence of living on earth is because of life existing on earth is because of photosynthesis. So ultimately all the living forms depend on sunlight for energy. So if we call ourselves as uh, the products of solar energy, nothing wrong in that, because ultimately all the living forms depend on sunlight for energy. So either directly or indirectly. See, the plants are dependent on sunlight for energy, and they are storing that energy in food. From the plants, it passes on to the herbivores. From herbivores, the food energy is passed. Food is it is consumed as food by the carnivores. That is how the energy is flowing along from the sun to the uh, topmost carnivore, that is a tertiary consumer. So it is flowing through these trophic level, various trophic level, and as it passes from one trophic level to another trophic level, there is only 10% of energy is passed. If a plant is absorbing uh, 10,000 joules of energy. So when it passes to the primary consumer, that is a herbivore, it would just be 1,000 joules. That is 10% law is there. So only 10% of energy is passed on from one trophic level to another. So you can just understand about how the sunlight or solar energy is playing an important role for the energy circulation, flow of energy in the ecosystem. Next academic year, we'll be studying about that in detail, but at present, our focus is on photosynthesis. Okay. See, photosynthesis, what is the importance of photosynthesis? It is the primary source of food on earth. So producers, they are the source of food. It is the primary source of all food on earth. It releases oxygen into the atmosphere. So this respiratory gas, the breathable gas. So oxygen is cycled by the photosynthesis process. Oxygen is liberated into the atmosphere. Okay. So experiments related with photosynthesis. If you look at the backdrop of the slide, this is a leaf which is coleus leaf. We grow it as an ornamental plant. It is a variegated leaf. You can find various colors on the same leaf. Okay, from black to uh, the purple to green. So you find various colors on the same leaf. So this is a variegated leaf, coleus, which you might have been growing as an ornamental plant. So we have an experiment with a variegated leaf. So it's better you are aware about this variegated leaf with different colors. Even the croton leaves, you might find different colors which are scattered within the leaf. Okay, we grow them as ornamental plants because of their colorful leaves. So the experiments related with photosynthesis, historically, how we came to know that the raw materials for photosynthesis are carbon dioxide, then they require photosynthesis liberates oxygen. Okay, all this was discovered step by scientists by various experiments. Let us study some of these experiments. Some of the experiments related with photosynthesis. So this variegated leaf experiment is there. So take a variegated leaf, any variegated leaf like coleus or croton, so which is or leaf partially covered with black paper. If you don't have any variegated leaf, you cover the leaf with partly with black paper. So and expose them to light. Test the leaves after 1 hour or 2 hours of exposure to light. Test the leaves for starch. It shows that photosynthesis occurs only in green parts of the plants in presence of light. So what is the test for starch? Iodine test. When you do this iodine test, you find that the starch would be synthesized only in the green parts of the plant. So that is where the reaction iodine test will work. It shows that photosynthesis occurs only in green parts of the plants and in presence of light. So this is the variegated leaf experiment. Then to exhibit carbon dioxide is required for photosynthesis, they conducted this Cole's half-leaf experiment. As per this, there is a split rubber cork in which half of the leaf is kept in a beaker containing KOH solution. So potassium hydroxide solution is there. So a part of the leaf is it can be even placed in a test tube, or as here it is in a beaker reagent bottle. They have done that. So a part of a leaf is enclosed in a test tube. Half of it is within the test tube or this reagent bottle, which contains KOH soaked cotton or KOH solution. So KOH solution, they have that capability of absorbing carbon dioxide. That is why we are using this potassium hydroxide solution, so that within this reagent bottle, all the potassium carbon dioxide that is present is removed. So half of the leaf is exposed to atmosphere, half of the leaf is within this reagent bottle or test tube containing KOH solution. So there is no carbon dioxide here, but light is available because the bottle is transparent. Okay, so they allow the setup for 1 or 2 hours. Later, they take the leaf for presence of starch. Now, this leaf is detached and it is tested for starch. The exposed part, that is the part which was exposed to the atmosphere, so they showed positivity for starch, and the portion which was within the test tube or reagent bottle, the half leaf which was within the portion within the reagent bottle, so they did not show any uh, test for starch was not indicated. The exposed part shows positive for starch, and the portion within the reagent bottle or the test tube, they showed negative for starch. This proves that carbon dioxide is a necessity for photosynthesis. So this half leaf experiment proved that carbon dioxide is a necessity for uh, the photosynthesis to take place, an important raw material for photosynthesis to take place. take place because the leaf which was exposed to sunlight but had no carbon dioxide could not synthesize starch, whereas the leaf which was exposed to sunlight and was exposed to carbon dioxide, they showed synthesis of starch, and when tested with iodine they reacted to it. Okay, so they showed positivity for starch test, whereas the leaf which was within this reagent bottle or test tube, they did not exhibit this positive. They were negative for starch test, so they did not react with iodine. Okay, so then the experiments up by Joseph Priestley, the person who discovered oxygen. Okay, so that is how he showed that we all require one of the elements in the air, which if it is present for breathing, we require that. If it is present, we are alive, and if we do not have that, so uh, we are going to die. So he showed that during his through his experiments, and he discovered this oxygen, an important aspect of living organisms to survive. We breathe in oxygen, and that is an important role for our survival, and the atmosphere has this oxygen. So this discovery was done by Joseph Priestley. So you should remember the important experiment done by Joseph Priestley, the scientist who did that. So Priestley performed experiments to prove the role of air in the growth of green plants. So he discovered oxygen in 1774. Okay, he observed that he did this experiment, bell jar experiment. In the first experiment, bell jar experiment, he had a lighted candle and a rat. So after 1 hour or after a few hours, he noticed that the candle flame got exhausted as well as the rat died. Okay, so the next experiment, bell jar experiment, he had the same candle, illuminated candle, rat, and a potted plant, a healthy potted plant, and he used this bell jar. Bell jar is transparent. Okay, so they are exposed to oxygen. He noticed that the candle flame also remained illuminated for a long time. It did not get exhausted, and the rat also remained alive. So, from these two experiments, Joseph Priestley was able to come to a conclusion that plant was liberating certain thing which was necessary for living organisms to survive, as well as the candle to have a uh, eliminated for such long time, which was not possible in absence of plants. So what exactly happened was when the rat and the lighted candle were placed in the bell jar, so they exhausted all the oxygen that was there. After them exhausting the oxygen that was present, so it was filled only with carbon dioxide. Carbon dioxide exhausted this flame. So they switched off the carbon flame of the candle, as well as because of lack of oxygen, the rat died. Okay, when the plants were placed, and the same experiment was repeated with a potted plant, a healthy potted plant, rat, and then lighted candle. They remained lighted, and the rat remained healthy for long duration of time because the plants liberated oxygen, which was the one of the important factor for anything to be lighted or burned. Okay, so the candle remained lighted for a long time because of the presence of oxygen, and the rat was also alive because of the presence of oxygen. So the plant should be liberating certain respiring air for long duration of time. That is what his experiment he came to a conclusion about. So Joseph Priestley observed that a candle burning in a closed bell jar, it got extinguished. Similarly, a mouse suffocated in a closed jar. So he concluded that a burning candle or a breathing animal damaged the air. So he had not yet named it as oxygen. So he concluded that a burning candle or a breathing animal damaged the air. So he later placed a mint plant in the bell jar, pudina, mint ones. He found that the mouse stayed alive and the candle continued to burn. He hypothesized, from this, he came to a hypothesis that Plants restore to the air whatever breathing animal and burning candles remove, that is oxygen. So Joseph Priestley came to an hypothesis, and he hypothesized that plants restore to the air whatever breathing animals and burning candle remove. That is, they are going to replenish the air with oxygen. So then the experiments by Jane House. Okay, so he conducted the same experiment by placing it once in the dark and once in the sunlight. So he was able to show that sunlight is essential for the plants to purify the air. So House, he showed that for the plants to purify the air or for the plants to liberate oxygen, so sunlight was necessary. See, this simple experiments made us develop on the concept of photosynthesis. The small experiments or simple experiments conducted by various scientists, it gave us a concept of photosynthesis. It was not just that we came up with this idea of photosynthesis on a single day. See, sunlight is required for photosynthesis was shown by Jane House that sunlight, when sunlight is there, the the material essential for purifying the air is liberated by the plants. When sunlight isn't there, the material required for purifying the air is not liberated by the plants. So that is what Jane House showed by conducting the same experiment by placing them in dark and in sunlight. He showed that Jane House showed that sunlight is essential to the plants for purifying the air. He repeated this experiment. Jane House, he repeated this experiment with an aquatic plant. It showed that in bright sunlight, small bubbles were formed around green parts, while in the dark, they did not do it. So he showed that in bright sunlight, small bubbles were formed around the green parts. Okay, while in the dark, they did not liberate these bubbles. So those bubbles are nothing but oxygen. Later, he identified these bubbles to be oxygen. Thus, he showed that only the green part of plants release oxygen. Okay, so Indian House, he also repeated this experiment with an aquatic plant. So it showed that in his experiment, he was able to show that in bright sunlight, small bubbles were formed by this. You can notice this oxygen being released by this aquatic plants. Small bubbles were formed around green parts, while in the dark, they did not liberate oxygen. Later, he identified these bubbles to be of oxygen. Thus, he showed that only the green part of the plants release oxygen. So, this is one of the work of Jane House. Then experiments by Julius von Sachs. So Julius von Sachs, we have already studied his contribution to the mineral nutrition, isn't it? So hydroponics, Julius von Sachs hydroponics experiment, we are already aware about. The same scientist, he proved that glucose is produced when plants grow and it is usually stored as starch. So he also proved that chlorophyll is located in special bodies called as chloroplasts. So the cell organelles called as chloroplasts, chlorophyll is present in special bodies called as chloroplasts. Glucose is made in the green parts of the plant. He also showed that glucose is made in the green parts of the plant. See, these were all the experiments by Julius von Sachs, where he proved that glucose is produced when plants grow and it is usually stored as starch. So glucose is produced whenever plants grow, and they are usually the reserve food material is stored as starch. So chlorophyll is located in special bodies called as chloroplasts, the cell organelles called as chloroplasts. Glucose is made in the green parts of the plant. So these were all the discoveries of Julius von Sachs experiments. So then experiments related with photosynthesis was one more experiment is T.W. Engelman. So this person is the Engelman. So 1843 to 1909. So this person performs certain experiments related with photosynthesis. So he split the light using a prism into its spectral components and eliminated a green algae, cladophora, placed in a suspension of aerobic bacteria. So the bacteria were used to detect the sites of oxygen evolution. So the aerobacteria grew only in the sites where oxygen was being liberated because aerobacteria required oxygen. Okay, so you use this green algae cladophora, which is spiral in shape. So he observed that bacteria accumulated mainly in the regions of blue and red light of the split spectrum. So it was the he was the first to describe action spectrum of photosynthesis. It resembles the absorption spectra of chlorophyll A and chlorophyll B. So he was able to show that the bacteria accumulated, see, whenever they were exposed to a waveguide of light that he had obtained through a prism, he was able to show that in the cladophora they grew only in the blue and red light uh, because there was accumulation of aerobic bacteria only at the blue and uh, red light spectrum of light. So he was of the conclusion that oxygen was liberated only at these blue and red wavelength of the spectrums. Look at these experiments, how simple experiments they were, but they gave us a lot of knowledge on photosynthesis. So it was the first described action spectrum of photosynthesis. It resembles the absorption of chlorophyll A and B from the atmosphere, the wavelength of light. Okay, so the action spectrum of photosynthesis was identified by his experiment, Engelmann's experiment. So he placed green alga cladophora in a suspension of aerobic bacteria. The bacteria were used to discover the sites of evolution. So he noticed more bacteria in the blue wavelength of light and red wavelength of the split spectrum. So by the middle of the 19th century, it is discovered that plants use light energy to make carbohydrates from carbon dioxide and water. So the plants, by the middle of the 19th century, it was already discovered that plants use the light energy to make carbohydrates from carbon dioxide and water. So the empirical equation of the process of photosynthesis is carbon dioxide plus water in presence of light and chlorophyll pigment. They were able to produce glucose and oxygen was liberated, where CH2O represents a carbohydrate. Okay, so they have not given it as glucose, it is just a carbohydrate general formula. C6H12O6 it has to be. Okay, it represents the carbohydrate glucose. So this overall reaction they were able to come to a conclusion with. Then there was experiments by Cornelius van Niel. So he was a microbiologist. He conducted some studies in purple and green bacteria. So you can notice this in the culture media, the purple and green bacteria. He demonstrated that photosynthesis is a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbohydrate to carbohydrates. So this can be expressed by 2H2A plus carbon dioxide. So this the source of this uh, hydrogen was later discovered to be water, but how they identified that there should be hydrogen obtained from a suitable oxidizable compound which reduces carbon dioxide to so glucose, that is a carbohydrate, and also there is a liberation of oxygen. Okay, so plus formation of water. So that was what was Cornelius van Niel's experiment. In plants, water is the hydrogen donor and it is oxidized to oxygen. In purple and green sulfur bacteria use hydrogen sulfide as a hydrogen donor. So the oxidation product is sulfur or sulfate and no oxygen is produced in this purple and green sulfur bacteria. So that is what you notice. They underwent chemosynthesis. So purple and green sulfur bacteria, they obtained hydrogen from hydrogen sulfide donor, whereas in plants, Water is the hydrogen donor and it is oxidized to oxygen. So the oxidation product in purple and green sulfur bacteria is sulfur or sulfate, and no oxygen is produced here. Thus, he was able to come to a conclusion. Cornelis van Neel, he came to a conclusion that oxygen is evolved by green plants comes oxygen which is evolved by green plants comes from the source of water, not from carbon dioxide. This was later proved by using radioisotopic techniques. Therefore, the overall reaction for photosynthesis is this is the overall reaction for photosynthesis. 6 molecules of carbon dioxide plus 12 molecules of water in presence of light and chlorophyll pigments. They are going to produce a single molecule of glucose plus 6 molecules of water and 6 molecules of oxygen is liberated. So this is the overall reaction of photosynthesis that was obtained after the work of Cornelius van Neel. So next we'll be studying about the photosynthesis site and pigments. So which are the sites of photosynthesis, chloroplast, and what are the pigments involved like rubisco, phosphoenol pyruvate carboxylase or PEP carboxylase. So rubisco is ribulose biphosphate carboxylase oxygenase enzyme. Okay, so we'll be discussing about which is the site of photosynthesis and what are the pigments involved. See, photosynthesis takes place in green leaves and it is also taking place in other green parts of the plant. Okay, so chloroplasts are present in the walls of mesophyll cells of leaves. So, leaf is a kitchen of the plant. You know that major photosynthesis takes place in the leaves. So, leaves will be having the mesophyll cells. This mesophyll palisade or spongy chlorenchyma, they are richly filled with chloroplasts. So chloroplasts are present in the walls of the mesophyll cells of leaves. So this chloroplast helps to get optimum quantity of incident light. So they try to absorb as much light as possible. So it helps to get optimum quantity of incident light. So chloroplast contains a membranous system which consists of grana, stroma lamella, and fluid stroma. So the chloroplast consists of a membrane system which consists of grana, stroma lamellae, and fluid stroma. So these are the things that are present within a uh, chloroplast. So we have already studied the ultrastructure of chloroplast. They have 2 layers, outer membrane, inner membrane. Then they have this uh, coin-like structures called as thylakoids. Thylakoids are arranged one above the other like a pile of coins, and we call this group as granum. Okay, so the grana are interconnected by this fret stromella, or we also call it as the stroma thylakoids. Okay, fret, or we can also call it as stroma lamellae. So then they have this fluid part within the chloroplast, which we call it as stroma. See, the cyclic and non-cyclic photophosphorylation, it takes place in the thylakoids, and the Kelvin cycle takes place in the stroma of the chloroplast. So chloroplast present in the walls of mesophyll cells of leaves, it is helping to get optimum quantity of the sunlight, the incident sunlight. So the chloroplast contains a membranous system. It consists of grana, that is, these thylakoids arranged one above the other to form a pile of coin-like structures. Then this granum are interconnected by stroma lamella, and they have a fluid part called as stroma. So the fluid stroma. Each granum is a group of membrane-bound sacs called thylakoids or lamellae. They contain leaf pigments. So they are going to contain the leaf pigments like chlorophyll A, chlorophyll B, xanthophyll, carotenoids. So the membrane system, they are going to trap the light energy and they help in synthesis of ATP and NADPH. What is ATP? Adenosine triphosphate. So the membrane system helps in trapping the light energy and they synthesize adenosine triphosphate, and NADPH, that is nicotinamide adenosine uh, dinucleotide phosphate, reduced nicotinamide, because they have H, it indicates reduced nicotinamide adenosine dinucleotide phosphate. It is called the light reaction. In stroma, the enzymatic reactions incorporate carbon dioxide into the plants for synthesizing sugar, which in turn forms starch. It is called dark reaction. So it dark reaction does not mean that they occur in darkness or that they are not light dependent. Okay. So the pigments which are involved in photosynthesis are, so pigments are substances that have ability to absorb light at specific wavelengths. So chromatography, the paper chromatography, probably if you had practicals, we would have at least demonstrated this paper chromatography, which shows the separation of the pigments. We crush the leaf and obtain the extract and use a what do you call it as the ascent solution. Okay. And when we dip it into that solution, there is separation of these pigments. So we call this as paper chromatography we use for the separation of pigments. So you can see the chlorophyll B, chlorophyll A, and xanthophyll and carotenoids, the orangish color, which have all got separated by this paper chromatography. So that we would have demonstrated it in the lab, but unfortunately we have not done that. So labs aren't going about. So pigments are substances that have the ability to absorb light at specific wavelengths. So already in one experiment, we have proved that in red and blue wavelength of light, maximum photosynthesis is taking place. And how we were able to show that was by the evolution of oxygen was maximum. When the evolution of oxygen is maximum at this wavelength of light, it indicates that photosynthesis is also maximum. How did we come to know that they are evolving maximum amount of oxygen? Because the aerobic organisms were concentrated at blue and red wavelength of light. Okay, so the chromatography shows the following leaf pigments in a chromatogram. So chlorophyll A, so which is bright or blue green. You can notice this bright colored chlorophyll A or blue green. Chlorophyll B, it is yellowish green. Xanthophyll, it is yellow. Carotenoids, they are yellow to yellow orange. So dark yellow to yellow orange. So the carotenoids are usually orangish yellow. Okay, these are the pigments that are found there, and they have ability to absorb light at different wavelengths. Chlorophyll B, xanthophyll, and carotenoids are accessory pigments. The major photosynthesis is brought about by chlorophyll A. So chlorophyll B, xanthophyll, and carotenoids are the accessory pigments. So the functions of accessory pigments are they absorb light at different wavelengths and transfer the energy to chlorophyll A. So they protect chlorophyll A from photooxidation. So the different pigments, that is chlorophyll B, xanthophyll, and carotenoids, they absorb light at different wavelength and supply it to chlorophyll A. At the same time, they protect the chlorophyll A from the photooxidation process. So the pigments involved in photosynthesis, you can notice this graph which shows the absorption spectrum of chlorophyll A, B, and carotenoids. So when you see this graph, you can notice that the absorption spectrum and action spectrum coincide closely, showing that photosynthesis is maximum at the blue and red regions of the spectrum. So at the blue and red regions of the spectrum, the photosynthesis is maximum. So we have to notice that particular feature of it. This graph also shows that chlorophyll A is the chief pigment associated with photosynthesis. So let us understand here the various um, graphs showing the pigment and accessory pigment. So the graph showing so in the x-axis we have taken the different wavelengths of light that can be available for a chloroplast. So the absorption of light by the chloroplast pigments. So you can notice where there is maximum absorption. So there is maximum absorption at the blue and red wavelength of light. So the chlorophyll A, the graph also shows that chlorophyll A is the chief pigment which is associated with photosynthesis. So graph showing action spectrum of photosynthesis. So when you compare the action spectrum and absorption spectrum, they coincide very closely, showing that photosynthesis is maximum at the blue and red region of the light. So you can also notice that the action spectrum and absorption spectrum, they are going to coincide closely at the blue and red region of the spectrum. So the graph showing action spectrum superimposed on absorption spectrum of chlorophyll A. And you can notice here they have the uh, maximum uh, is seen. The absorption spectrum and action spectrum coincide closely at the blue and red region of the spectrum. So that is what you have to remember. Then the photosynthesis site and pigments. So the photosystems or pigments are organized into two photosystems. So we call them as photosystem 1 or PS1 and photosystem 2 or PS2. So these are named in the sequence of their discovery. So photosystem 1 and photosystem 2 are named in the sequence of their discovery. Each photosystem has a chlorophyll A and accessory pigments bound by the protein. So each photosystem is having a chlorophyll A pigment and also accessory pigments. They are all bound by proteins. So you can notice this photon light harvesting complex, a photon of light when they fall on them. So the reaction center, so the primary acceptor, so the electrons are being liberated by this pigment molecules. So photosystems, all pigments except one molecule of chlorophyll A form a light harvesting complex, LHC, or we also call it as antennae. All the pigments except one molecule of chlorophyll A, they form a light harvesting complex, which we also call it as LHC or antennae. Single chlorophyll A, they act as a reaction center. So the single chlorophyll A, they are going to act as the reaction center. In photosystem 1, the reaction center absorbs light at 700 nanometer. So photosystem 1 is also called as P700. In photosystem 2, the reaction center absorbs light at 680 nanometer, and it is also called as P680. So P680. So the reaction center in photosystem 2, they absorb the light at 680 nanometer. So we call the photosystem 2 as P680. The next concept is the light reaction or photochemical phase. So I've already discussed that photosynthesis can be studied under two stages. So the light reaction and dark reaction. So light reaction is also called as photochemical reaction. Dark reaction is also called as biochemical reaction. Light reaction, light is required for this reaction to take place. Photochemical reaction or the biochemical uh, reaction, that is the dark reaction, so they are light independent. They neither require light nor the light need not be absent, it need not be dark. Okay, they are light independent. Light doesn't have any effect either by its presence or absence. Light doesn't have any effect on it. That is why we call it as dark reaction. So these two are taking place in light reaction. important steps that take place is synthesis of ATP and NADPH2. Then you are also noticing that there is evolution of the photolysis of water happens. That is, water splits up and they evolve oxygen. That is also taking place in the light reaction. So the liberation of oxygen, the formation of ATP, formation of NADPH2. These are the important steps taking place in the light reaction. So whereas in dark reaction or Kelvin cycle, there is fixation of carbon dioxide, reduction, and regeneration stages. So carbon dioxide is fixed to form the glucose. That is the important steps that are taking place in the dark reaction or the biochemical phase. So let us study initially about light reaction or photochemical phase. So light reactions includes absorption of light, water splitting, oxygen release, formation of ATP and NADPH. See, ATP is the energy currency of the cell. In almost all the living organisms, ATP is the energy currency of the cell. NADPH is energy rich molecule. See, one NADPH2 is equivalent to 3 ATP. So when an NADPH2 undergoes this electron transport chain, they are going to liberate 3 ATPs. So it is an energy rich molecule. So when NADPH2 is oxidized, they are going to liberate 3 ATP. But the cells can recognize energy only in the form of ATP. In all the plants and animals, ATP is the energy currency of the cell. Adenosine triphosphate is the energy currency of the cell. So the ATP is stored in NADPH2. NADPH2 is energy rich molecule. One NADPH2 is equal to 3 ATPs. When they undergo oxidation, one NADPH2 liberates 3 ATP. So ATP and NADPH2 are high energy chemical intermediates that we obtain during light reaction or photochemical phase. So light reactions, it involves light absorption, splitting up of water or water splitting, oxygen release, and formation of ATP and NADPH. If you look into this slide, so the light is going to it is going to be incident on the leaf. So they take up water and carbon dioxide and they are going to undergo photosynthesis. Oxygen is liberated. So the NADPH2 and ATP, the energy rich high energy chemical intermediates that are formed, they are utilized in Kelvin cycle. So Kelvin cycle or dark reaction. So where they are going to fuse the carbon dioxide. Carbon dioxide is fixed in Kelvin cycle, and these energy rich molecules are utilized to finally synthesize the glucose or sugar. Now let us understand the Z scheme of light reaction. Before that, the electron transport. So whenever photosystem 2, they absorb red light of 680 nanometer wavelength. So electrons are excited and transferred to an electron acceptor. So electrons are excited whenever the photosystem 2 absorbs red light of 680 nanometer. So the electrons are excited and they are transferred to an electron acceptor. So the electron acceptor passes them to a chain of electron transport system consisting of cytochromes. So this movement of electron downhill in terms of redox potential is downhill in terms of redox potential scale. So there is an acceptor of electron and donation of electron. Both are taking place. So they not only are acceptor of electrons, but they are also donor of electrons. So the electrons are transferred then from this electron transport system to the photosystem 1, okay, which has 700 nanometer wavelength of light. So simultaneously, electrons in photosystem 1 are also excited when they receive red light of 700 nanometer, okay, and they are also transferred to an another acceptor acceptor molecule having a greater redox potential. So these electrons are moved downhill to a molecule of NADP plus. As a result, NADP plus is reduced to NADPH plus H plus, that is NADPH2. So transfer of electrons from photosystem 2 to photosystem 1 and finally downhill to NADP plus. It is called as Z scheme due to its zigzag shape. So this is called as Z scheme due to its zigzag shape. So this shape is formed when all the carriers are placed in a sequence on a redox potential scale. So splitting of water, the water splitting complex in photosystem 2 is located on the inner side of the thylakoid membrane. So water is split into H plus nascent oxygen, that is O, and electrons. So two molecules of water, they are going to split as four protons plus oxygen molecule which is liberated and four electrons. One water is, it is nascent oxygen. So four electrons are liberated. So photosystem 2 can supply electrons continuously by replacing water from water splitting complex. Thus, photosystem 2 provides electrons needed to replace those removed from photosystem 1. So the protons H plus are used to reduce NADP to NADPH. So oxygen is liberated as a byproduct of photosynthesis. So this is what is taking place in this Z scheme of light reaction. Okay. So I'll just have this part of it. So the Z scheme of light reaction, what you notice is there is absorption of light, water splitting, oxygen is released, and there is a formation of ATP and NADPH2. So NADPH2 is a energy rich molecule. I told you that one NADPH2 is equivalent to 3 ATP. So during respiration, NADPH2, if they undergo oxidation, they produce 3 ATPs. So ATP and NADPH are high energy chemical intermediates. Okay, so the electron transport we were discussing and the Z scheme of light reaction. This is especially in non-cyclic photophosphorylation. You have the Z scheme. Okay, when photosystem 2 absorbs light of 680 nanometer wavelength, red light of 680 nanometer wavelength, electrons are excited and transferred to an electron acceptor. So the electron acceptor donates these electrons, which passes through a chain of electron transport system consisting of cytochromes. During this process, ATP is also synthesized. The movement of electron is downhill in terms of redox potential scale. So they are downhill. Why have we not written it uphill? They are downhill in terms of redox potential scale. So the electrons are transferred to the pigments of photosystem 1. Simultaneously, what is happening is electrons in photosystem 1 are excited when they receive a red light of 700 nanometer and they are transferred to another acceptor. So the electrons are transferred to another acceptor. So these electrons having a greater redox potential, these electrons move downhill to a molecule of NADP plus. As a result, NADP plus is reduced to NADPH plus H plus. So transfer of electrons from photosystem 2 to photosystem 1 and finally downhill to NADP plus is called the Z scheme. So how is this arranged as why it should be upward direction? It is based on the redox potential scale. So when they have a higher redox potential scale, it is uphill. When they have lower redox potential scale, it is downhill. So that is how we are obtaining this Z scheme. So transfer of electrons from photosystem 2 to photosystem 1 and finally downhill to NADP plus is called the Z scheme due to its zigzag shape. This shape is formed when all the carriers are placed in a sequence as per their redox potential scale. So all these are formed because they are placed as per their redox potential scale. So the splitting of water is an important thing that is happening. The water splitting complex in photosystem 2 is located on the inner side of the thylakoid membrane. Water is split at this photosystem 2. This is happening. Photolysis of water or water splitting complex. So water is split into, if it is a single molecule, it will be proton nascent oxygen. So when you're putting this within brackets, it implies nascent oxygen and electrons. It is unstable. Nascent oxygen is unstable. So if it is two molecules of water, they produce four protons and oxygen molecule is liberated and four electrons are liberated. So photosystem 2 can supply electrons continuously by replacing electrons from water splitting complex. Thus, photosystem 2 provides electrons needed to replace those removed from photosystem 1. The protons are used to reduce NADP to NADPH. Okay, the oxygen is liberated as a byproduct of, you can notice this water splitting happening here. So oxygen is liberated as a byproduct of photosynthesis. So this is what is taking place in the electron transport. The splitting up of water is taking place. And also you can notice photophosphorylation. ATP is synthesized in presence of light. Phosphorylation means synthesis of ATP by cells. In mitochondria and chloroplast, we call it as phosphorylation. Photophosphorylation is the synthesis of ATP from ADP in chloroplast in presence of light. So it is taking place in chloroplast in presence of light. Photophosphorylation takes place in two ways: non-cyclic photophosphorylation and cyclic photophosphorylation. So photophosphorylation is taking place in two ways: non-cyclic photophosphorylation and cyclic photophosphorylation, which we'll be discussing in the next class. Okay. Can you so with this, we'll stop with today's discussion. Do you have any doubts or clarification, dear students?